It's my privilege and my honor to be able to introduce our keynote speaker today. And I just want to spend a couple of minutes. I don't want to eat up too much of his time because it's already been long enough that I've taken. But I just want to tell you a few things about him. He is a professor of neuroscience and the director of the Kavli Institute for Systems Neuroscience at the Norwegian University of Science and Technology in Trondheim, Norway. He did most of his formative training at the University of Oslo with Per Andersen, which I think he shares in common actually with many people in this audience. He then did a postdoctoral fellowship with John O'Keefe and with Richard Morris at University of Edinburgh and UCL. And the work that he's going to talk about today, the large corpus of work that he has done in his career in collaboration with Edvard Moser, focuses on how spatial memories and spatial locations are encoded in the brain and the mechanisms that are required to formulate some sense of where you are and how you can navigate in space. Now, this work was incredibly influential and transformative. It earned him, Edvard Moser, and John O'Keefe the 2014 Nobel Prize in Physiology or Medicine. Some of our speakers' more recent work, which I hope he will have an opportunity to talk about today also, focuses on taking this premise of understanding neural computation underlying space and memory in the brain to trying to understand time and understand how time is also processed in the brain. And maybe there are shared mechanisms there, maybe there are differences. I think we'll have to wait to hear from him on that. But that's something I'm particularly excited to hear from him about, some of these new directions in the work. One of the things I want to mention about our speaker also is that if you have a chance to spend more than a couple of minutes with him, you'll realize something very, very special. Aside from his global renown and his accomplishments, he's also one of the most humble people I have ever met. And I think you'll know this just by talking to him for a few minutes. He's very generous with his time, with students, with colleagues, and I've always had a listening ear when I've tried to reach out to him and chat about data and science. I also want to take this opportunity to thank Neuralinks, who have sponsored this keynote lecture. Neuralinks and our speaker actually have kind of a history. They go way back. And this is something that I think is really just spectacular for us to be able to have their support for this conference, and in particular for this keynote lecture. Now, I know you're in for quite a treat. I don't want to take up any more of your time. So with that, ladies and gentlemen, please help me give a warm welcome to our speaker, Edvard Moser.
So thank you a lot, Mike, for the nice introduction. And thanks to both you and Manuela and everyone else who has organized and prepared this conference. I think the size of the audience, the number of people here testifies to...
there we go. Okay. You don't want to have that slide up the whole time.
Not only to the great work that is being done here, but also to the importance of this center in the history of modern neuroscience, and especially with a focus on learning and memory. And my congratulations especially to Jim McGaugh for starting all of this and for doing this for 35 years. So my talk will be, I was told explicitly when we started, when I prepared this, that this is a combined public talk and scientific talk, which is something that's really hard to achieve actually. But I will start out at primary school level in the beginning, and then I will go gradually up, and during the second half of my talk, I will move into unpublished territory and include some new principles of position coding in the entorhinal cortex, and then move over, as Mike said, to time, which is the most recent work, and which will serve as an introduction to another talk that my former Ph.D. student, Albert Zhao, will go into more detail on tomorrow. But let's begin with location and space. So I thought I could not be worse than both Jim and Lynn, who both had pictures of gull, so I'll do the same. And here's my gull brain. It shows the different faculties or abilities or properties and how they are labeled onto the brain. And I think both speakers made the important point that this had actually a tremendous influence on neuroscience. It set the stage and then was forgotten for many years, but actually today with trajectories into circuits and not only areas and principles for cooperation, collaboration, interaction between many cells, we are actually getting back to the point where we can start to understand some of the psychological functions that are enabled by the brain and especially the cortex. However, since the 19th century, concepts have moved forward too, and that's also one of the reasons now with more conceptual advances and better ideas and models for how the brain might work at the psychological level, we are actually making some advances. But there's more advance in some areas than in others, and one of those areas that over the last 40 years or so really have seen a lot of advance is our understanding of how space is represented. Because this is one of the first, in mammals, one of the first high-order non-sensory and non-motor functions that are really beginning to be understood in neural language in terms of how cells work together and how cells have different functions and how this is all put together to produce something that probably gives rise to our sense of location. So I want to start very simply, and now I will go to primary school level and simply ask what would it be like if we didn't have this ability to conceive of space and where we are in space. So I have an animation that we made for the purpose, not for this purpose, but I will show this, and that takes about 2 minutes. And so let's begin with this. Now sound should be on. Well, wait. Is sound on? Okay, try again. Nope. Well, I'll wait because sound is essential, really. It worked 1 minute ago. So.
Is this turned on?
I don't know.
Okay, let's go ahead.
Okay, try once again. There, good. Yeah, fantastic.
Turn it down if you need to.
Yeah.
How does life deal with space? It navigates. Life forms develop and change over time. Abilities and traits that prove useful for survival are retained across generations through the succession of species from the common ancestor to its progeny. These are the mechanisms of evolution. Natural selection has favored the species with the best ability to navigate. An organism that moves can escape from danger and find shelter. Navigation also allows us to actively find food. The safety of a flock, or a suitable mate. Scientists have discovered a navigation system in the brain that is common for mammalian species as diverse as bats, rats, mice, monkeys, and even humans. These discoveries suggest that this positioning system evolved from a common ancestor of mammals or earlier. We all share this system.
So, where is this system? Well, there are many parts of the brain are involved in space, but there is still, as we have learned earlier today, there are two areas that have received a lot of attention that are critically involved in representation of space. So that's the hippocampus and it's the entorhinal cortex. Is this the pointer? Let's see. Oh, yeah. Okay. So this shows the human brain, and this shows a rat brain. So this is all from collaborative work with Menno Witter. It shows the human brain in the red area, which is embedded under the deep in the cortex here, and the blue area here is the entorhinal cortex. In the rat brain, it's located somewhat differently, but very far back. The hippocampus here, entorhinal cortex here. And in these areas, they have turned out to be important, but because it is all much easier to investigate in animals than a major advance was made about 45 years ago, as you've heard already earlier in this meeting, when John O'Keefe and Jonathan Dostrowski started to record electrical activity or action potentials, spikes, from single neurons in the hippocampus of rats. So this shows a rat that is walking freely in a box, or it could be other types of apparati, mazes. But in any case, what John did was that he recorded activity from single cells and viewed those on a screen, on an oscilloscope, and stored them, and then found that single neurons in the hippocampus are responsive to the location of the rat. So I will illustrate this with a movie. Now we see the rat from above. The rat is walking in a box. The box is 1 meter by 1 meter. In the box there are occasionally thrown out crumbles of chocolate, which rats like. That keeps them walking around in the box and visiting every possible place. And at the same time, we are recording cells from the hippocampus. You will hear those soon as spike sounds or noise, sounds like noise. But each time there is a sound, a popcorn sound, then this cell is active. And you will notice that this cell, example cell, is active only at certain places in the box. So let's start the movie. And each time the cell is active or fires, you'll also see a red dot up on the screen. So you probably already now have noticed that this cell is active only at one place in the box, in this case in the upper left part, and otherwise the cell is very silent. This can also be illustrated with a heat map, a color code, where red is high activity, blue is low or no activity, and it turned out that then during the years to come after this discovery that different cells have different preferred areas in the hippocampus, and together it became clear that these cells cover the entire environment visited by the rats, and for these, based on these data, then John O'Keefe and Lynn Nadell suggested in 1978 that the hippocampus is actually the basis of a cognitive map, that, or a Tolmanian map. You heard in the morning about Tolman's contributions, a map that encodes spaces, locations in the environment, but also, more than that, also experiences associated with those locations. This was a major conceptual advance. It put things together, tied it up, as you heard this morning, a very chaotic literature. So during the next decades, a lot was learned about place cells, but there were a few things that still weren't clear. when we came into the picture after a 3 months visit with John, where he taught us all the essentials. So when we started up in 1996 in our own lab in Norway, there were several questions that were interesting, but one, and perhaps the most important one, was which hadn't been resolved. Where and how is this place cell signal generated? Because this, remember, this is not sensory cortex. So these signals, they have the properties that are as clear as you might see in sensory areas, because they really strictly respond to the location of the rat. But, as you all know, you don't have space sensors on your fingers, not in your ears, not in your eyes. So how is this generated? Where does it come from? So to a large extent, this is probably generated inside the brain, by the brain itself, based on, with the help of sensory inputs, but this was really not well understood. And one idea that was around in the 1990s was that if anything, this signal, if it wasn't created in hippocampus, it was at least enhanced quite significantly in the hippocampus. And because the hippocampus operates to a large extent like a circuit, a unidirectional circuit consisting of sub-areas that project from one to the other in a loop through the hippocampus, in, through it, and out. Then, and most of the cells have been recorded in CA1, which is one of the last stages of the circuit. Then it was believed by many people at that time that essential things happened in the earlier stages just before the area where most of the activity had been recorded. So an obvious thing to do when we started out was simply just to try to get rid of the inputs from CA3 that was postulated to be so important. And what we found, which actually was in agreement with earlier work using other methods from the McNaughton and Barnes lab, was that a lot of the activity actually survived. So this shows examples of 7 different cells from a recording when the CA3 is inactivated or lesioned. And you can see that these 7 cells, this is the box seen from above, and color indicates the activity of the cell. You see that these cells are still spatially selective. They still fire in certain areas and not in other areas. So although the spatial firing was not as strong, maybe, as it is in a control animal, it was still not really noticeably different. So that then led us to get interested in the entorhinal cortex, an area that feeds in most of the cortical input into the hippocampus. And by that time, we had strengthened connections with Menno Witter, who then participated in this study and was one of the world's experts on just this area. And this work, and we showed then here that with Menno that this was the case, that the place signal survived despite in animals where there was absolutely no input from the CA3 left, which we showed by using anatomical methods. So that led us to the entorhinal cortex, and we tried to record directly from that area together with Menno and with the students Marianne Fyn and Torkel Hafting. And we put then the recording electrodes into the dorsal part of the medial entorhinal cortex. And this dorsal part is the area that has the strongest inputs into the dorsal hippocampus, where almost all place cells had been recorded. So it was an obvious area to go to, but at that time, in that part of entorhinal cortex, I don't think anyone really had recorded yet. So it was new territory. And what happened was then that cells in that area had a different type of pattern. First of all, they were very strongly spatially modulated. So what you see here now to the bottom right is the box again. Now it's a bigger box, in this case a 220 by 220 centimeter large box. The gray trace is where the animal walked, so it shows the path of the animal over half an hour. And each black dot is where that one particular cell was active when the rat was running around. So what you can see is that that this cell, like other cells in the area, was active in certain places, but no longer just in one place, or maybe it was active in many places. And the other thing that you may notice is how regular this pattern is, which you can see when you put these lines on top, which I did in the left diagram here. That is actually a repeating triangular or hexagonal pattern that in many ways expresses a metric that was certainly not present in the place cell signals of the hippocampus. So apparently here we had another component of this spatial or cognitive map that contained information about distances and directions that were not so easily extractable from the hippocampus. So this is now 2004-05. So one of the things that became clear from the beginning is that the grid cells, there were many of them, and especially they were abundant in the superficial layers of the medial entorhinal cortex, which project into the hippocampus. But they varied in various ways, so they could have different phases, they could have different scales, or they could have different orientations relative to the environment. So phases means that the grid patterns are shifted in XY space relative to each other. So you see that illustrated here for a green grid cell and for a blue one. And you can see that they have different, the peaks of the grid pattern are at different places. Or they might differ in the scale, which you see here with the blue one compared to the green one. So you see the blue one has both larger fields and larger distances. And the third is then that they may be tilted relative to each other as well. So we asked then early on whether there is any organization according to these dimensions, and both yes and no. So first of all, for the face of the grid, there was no very striking organization of that. That means that wherever we recorded, and this illustrates our recording electrodes, we're using tetrodes, which I don't have to explain how that, I don't have time to explain how that works. But anyway, it picks up signals in a way that makes it possible to differentiate between cells and to isolate from each other. So here you have a blue cell, a green cell, and a red cell. And you can see that the grid patterns on the three cells, all of them have grid patterns, but they are shifted in XY space. And this is pretty representative of what you get in most places, so that it is similar to what in sensory or visual neuroscience often is referred to as salt and pepper organizations, pretty mixed. Whether that is totally mixed, that is still uncertain, and there are various indications recently that there may be some organization to it. There may also be not an equal distribution of phases, but by and large, every location is represented at every place, every anatomical location in the entorhinal cortex, which is very different from the spacing of the grid, because it was clear from the outset that spacing varied depending on how far up or down you are in the brain. So this is a side view of the hippocampus and entorhinal cortex. Hippocampus is this ear-like structure here, and the red structure here is the medial entorhinal cortex. If you start at the top, which you often refer to as the dorsal part, and then go down towards ventral or the bottom, what we typically see is that it starts out with only small-scale grid cells. Dots are small and very close to each other. So this is a box of 220 by 220 centimeters, and the distance here is down to something like 30 centimeters between each node. As you go down, this increases, and so you get into the middle here. It may already be a meter or more, and If you go even further, then it's difficult to assess because we don't have environments that are big enough, or didn't at least at that time. So there's a clear gradient, topographical gradient, where it begins with the smallest at the top and goes towards the largest at the bottom. So this can be organized in many ways, but one important question was whether is this a continuous gradient where you go smoothly from smallest to largest, or are there actually discrete steps? So does this depend or consist of subnetworks that each have their own scale? And certain ideas about how grid cells arise actually require the latter, so that we did look more into this, and this is work with Tore and Hannes Stensole in about 2012, who were able to record up to almost 200 grid cells from the same animal, which at that time was quite unique. And by doing so, they were able to then plot the scale of grid cells from the same animal in one diagram. So what you have here is on the x-axis, you have dorsal to ventral, so top to bottom in the medial entorhinal cortex. And then on the y-axis, you have the scale of the grid, or the distance between the peaks. And then each dot is one cell. And what you can see is, first of all, as I told you, as you start from dorsal and go to ventral, then the scale generally gets larger and larger and larger. But what you also see is that it is a step-like increase where actually just a small number of scales present, and almost every cell can be put into one of these steps. So these steps, we call them modules, and we call them module 1, the smallest one, and then module 2, 3, and module 4. So it turned out they even have a certain relationship, so that you, when we asked what is the factor that you have to multiply M1 with in order to get M2, how much do you have to multiply M2 with to get M3, and so on, turns out that it's actually a constant factor. And in this case, under those conditions, in rats, it was approximately, or on average, the mean was 1.42. Of course, there's a lot of variation, but still, the scale factor is the same, so that you can actually describe the levels of the grid cells, or the modules of the grid cells, as organized in a geometric progression. So, and what's the advantage of that? Well, that is still not clear, but it has been hypothesized, at least by various people, that this might be the best way to organize grid scales if you want to represent space in the possibly most efficient manner with the fewest number of cells. So I want to emphasize at least one major difference between the place cell map and the grid cell map. So we go back to the place cells now. You heard also again from the morning talks and also from Carol's talk that one property of place cells is that they remap, as we say. That means that you have different maps or different combinations of place cell maps in different environments. So we can also say that the map is high dimensional, because it just what this means is that the maps are uncorrelated. They are as different as possible, and this was shown already by starting with Muller and Kubi, and then has been developed by many labs over the years. But I like to show this experiment that we did quite recently, because we demonstrated effect in as many as 11 different recording rooms. So here's a picture of 11 labs where rats were tested. And I like to show them because those labs are so similar that I can't tell the difference. There's no way that I can say the difference between lab number N8 and lab number N2, for example. But The question is whether rats are able to. So what Charlotte Alme in our lab did was that she recorded place cells, many place cells from the same rat in a sequence where the rat was tested sequentially in all these rooms. One familiar room, which has the label F, and then 10 different novel rooms where they were exposed for the first time, labeled from N1 to N10, and then asked whether place cells in those rooms, are they similar? Is it one map that is carried over, or as we expected, because of their ability to remap from one room to other, that they are uncorrelated? So what we found in this experiment is that all combinations of maps in all of these rooms are actually as different as it is possible. So what you see here is, first of all, maps from place cells in, so this is cell number 1, cell number 2, 3, 4, and so on until cell number n, and then they are correlated using a population vector approach. And this is a correlation matrix, and this is all the different rooms on one axis and all the different rooms on the other axis, and then the color indicates the correlation between the maps. And of course, along the diagonal, when you correlate the room with itself, you get a correlation of 1, so that's no surprise. The same recording correlated with itself. But on all other combinations, you see that it's in the deep blue range, which means essentially what you would get by chance. It's absolutely not different if you just shuffle the data completely, except for a very few places here, which all are marked by a star or an asterisk. And those are the instances where the room was actually repeated, a second exposure to the same room. And then, so that shows that it's not just the fact that a new map is pulled up each time, but it actually, the same map is re-expressed when they go back to the environment. But otherwise, those maps of place cells are as different as they can be, which is probably quite useful and what you want to have in a structure that stores memories, including spatial memories for many places. You don't want to mix them up, you want to keep them separate. So this is in line with all the work that has implied a role for the hippocampus in memory, which we heard more about this morning. So this is quite different from what we see in the entorhinal cortex. Because in the entorhinal cortex, there is not this scrambling between environments. So I'll illustrate this first again. Now, same type of approach. You compare two different rooms. This is room A, this is room B. In one of the room, it was a circle. In the other, it was a square. But anyway, many cells were recorded at the same time in both rooms. And then cross-correlated, so similar correlation between the maps for different environments. And this shows the result for 1, 2, 3, 4, 5, 6 cells. And the first row shows when you correlate the map with the recording in the same environment, so A times A. And of course you get a grid map because there's no reason why it should change. And of course you also get a peak in the center because there's no reason why the map should move. But if you now correlate the A versus the B, you also get a grid map, except that the map is shifted slightly to the right in this case, which means that the cell has the same pattern, it's just slightly displaced in one direction. But the important thing here is that this shift is expressed in every single cell that was recorded. They all show the same shift, which then means that actually here you have one map that is just shifted in X or Y, or maybe it could even be rotated, but it's the same map. And this is even the case if you put all the cells on top of each other and cross-correlate them all together. Again, you get the same map, but just slightly shifted. So what this suggests is that it's really just one map that is used over and over again, at least as long as you stay within one of the modules. So at that time, we didn't know about modules, but there's still reasons to believe that most of the cells were from one single module. So based on those data, which is a study essentially all from a study by Marianne Fien in 2007 and in a collaboration with Alessandro Treves, then we asked more recently how much is this a single map that actually is expressed even when the rat has no behavior, for example, when it's sleeping and not walking around? So this is a study that we did and actually has been shown. The same result has been shown also in Laura Colgin's lab, where they did the same at the same time with exactly the same result. So then I believe it. So what we found, and this is Richard Gardner's work in our lab, what he did was first to compare pairs of cells that were in phase. That means that the grid pattern is more or less overlapping. So you see two grid cells here, and you can see that the peaks are more or less in the same place. You can also see that from the color, but maybe easier to see here. So these are examples of two cells that fire in the same place when the rat is awake and walks around in the box. And as you would expect, if you cross-correlate those two cells in time, show what is the probability of cell 2 to fire when cell 1 is active, you get a strong peak around 0, because when one fires, then the other fires too. That's all as expected. But if you then record from the same cells in sleep, in slow-wave sleep, you get the same peak. So that means that those cells that fire together in the wake state also fire together in sleep. And conversely, if the cells are out of phase, if they have their dots or peaks at different places, then and fire out of phase in the wake state, then they also fire out of phase in sleep. So it's the same thing, and if you do this now for 1267 combinations or pairs of grid cells, what you find is that, and you can plot that then in one line per cell pair, and then now you can transform this one, this plot to color, so that yellow is high cross-correlation and black is low. So what you then find is that those pairs that have a high cross-correlation in the wake state when the rat is walking in this open field environment, they are also the ones that have the highest in sleep, and those that have the lowest correlation in the awake state when it walks in the box are the ones that have the lowest correlation in the sleep state. And the same applies also when in a different type of sleep, REM sleep, which in humans corresponds to when we dream. You say it's the same thing again, a bit more noisy because there's much less data from that, but what this essentially shows is that confirms the suggestion that this entorhinal map is really low dimensional, or has only one or at least only a few ways to express itself, very, very differently from the hippocampal maps, which can appear in all kinds of combinations. And this is exactly as would be predicted by attractor type models for grid cells, or models that propose that grid cells actually arise as a consequence of how the network is wired together, and these connections and these interactions, they are present also in the sleep state, even if these animals don't walk around. So with that, I then, that is a little bit of introduction about grid cells. I should also mention, though, that there are other types of cells, which already, some of you may have heard that from the symposium earlier today, and also was also mentioned in earlier, but not the least, the head direction cells. So head direction cells are cells that fire in when the animal's face is pointing only in a certain direction. These cells were discovered by Jim Rank, and then Jim Rank with his student Jeff Taube followed up and showed them. They found them originally in the dorsal presubiculum, which is adjacent to the dorsal medial entorhinal cortex, and but it turned out then that they are very abundant also in the medial entorhinal cortex. So this shows again a side view in a rat brain, and the area between the two red areas, lines here, is medial entorhinal cortex. And what you see here is that these cells, they don't really have these grid dots like you saw in the other cells, but what they have, as you see here, and this is a polar plot that shows firing rate as a function of direction of the rat's head. You can see that this cell, for example, only is active when the rat has its head pointing in the left or west direction. This cell is only active when the rat is walking from bottom right to top left. So these are strongly directionally tuned cells. Some of them are very, very sharply directionally tuned, others a little bit broader. And some of them can also be head direction cells and grid cells at the same time. There are also other cells, border cells we name them, cells that fire exclusively when the rat is walking along one or several borders of the local environment. So here again you see the box from the top. Color indicates activity or firing rate. Red is the highest rate, and you can see an example here of a cell that fires only when the rat is on the right part of the box, and that happens even if you stretch the box either in the horizontal or in the vertical, or either in the X or in the Y direction, still just fire at or along that particular wall. This shows the same cell in a different room. So now the cell chooses the left wall instead. And what you see here in the middle is that if a wall is inserted in the middle here, then the cell actually fires along that wall too, and on the corresponding side. So on the right side here, just as it does on the right side for the peripheral wall. So these, it's a very different type of cell. A grid cell is never a border cell, and a border cell is never a grid cell, at least not in our hands. So different classes of cells, and as some of you may have heard in the morning, they respond differently to sensory inputs, visual inputs versus locomotion, for example. But these cells coexist, they're intermingled in the superficial layers of the entorhinal cortex, and very closely associated also with the head direction cells, which are also there but shifted slightly more into the deeper layers. And more cells that many of which are actually, you heard about, some of you may have heard about speed cells. These are cells that don't really, as you'll see, the 12 example cells here, they don't really have a preferred location of firing. The color code or the heat maps here show that they are active anywhere in the box. But what the line diagrams here show is that their activity is strongly correlated, linearly correlated with the firing rate or with the speed of the animal. And that's also clear from the examples here. 7 different cells shown in different color. On a background of the speed of the rat. So the speed is shown in gray over a period of 2 minutes, and then the color shows the firing rate of the cell. And you can see, for example, if you focus on the yellow one here, you can see how closely the cell's firing rate actually follows the speed of the rat. It's extremely closely tied to the speed. So, and the existence of these cells is also kind of predicted, because if self-motion is necessary for updating these cells, if these cells actually use path integration to decide where they fire, then motion in self-motion or speed input is just as essential as the direction input. And finally, there are many cells that actually have spatially localized firing fields that aren't anything really. They're not borders. They're just blobs of firing at particular locations. Of course, they could be grid cells that, for some reason, either they have so large grid patterns that you just see one peak, or the other grid fields are so low in rate that you don't see them. But nonetheless, they are hard to explain, and there are many, many of them. They have been around for a long time, but until quite recently, At least I thought they were mostly just garbage, cells that you couldn't really put into any category. But I changed my mind slightly when Chenglin Miao in our lab showed that they are modulated in a different way than many of the other spatial cells. So what he found, which you can see an example of here, is that if you block somatostatin expressing cells, interneurons, and you silence them by using chemogenetic methods, then what you find, as you see in the middle column here, is that those cells under blockade of these somatostatin expressing interneurons actually get much more dispersed firing, and then when the drug is out of the body again, then they go back to what they were. So this happens only to these cells. So a grid cell, for example, would not respond to that treatment. And conversely, if you block another type of interneurons, parvalbumin type of interneurons, then there's no effect on these cells, so as you can see here, but there is a strong effect on grid cells instead. So it seems like these are actually different classes of cells that are modulated separately. So all in all, this then brings me back to the movie where I started, which suggests that that these cells they are widely expressed. Actually, they are present in many species. They were found first in rats and came then in mice, not surprisingly. But then they were discovered in bats, in the Ulanovsky group. And bats are on a completely different branch of the mammalian evolutionary tree. And then grid cells, or at least grid-like cells, were found in monkeys with buffalo's work, and then finally from Josh Jacobs and Itzhak Fried in humans. So the fact that they are spread around among mammals probably suggests that they arose quite early and are present widely among mammals. So that applies not only to grid cells, but it applies at least to several other types of cells, like border cells and head direction cells. So that was my long introduction, but I did want to save some time for a few new things. So one of the first questions, and it probably come up already to everyone who is here who is not working in the field. So may I ask, why do they only test these animals in these empty boxes? Because rats don't really walk in empty boxes in their natural lives. So how about more realistic environments? Realistic environments, how are they different from empty boxes? Well, at least they contain objects. There are things in the environments. So there is some precedence from other approaches that suggest that rats or animals may actually use objects for navigation. And that includes both behavioral work, and especially the work of Tim Collett, which is illustrated here. And just the 5-second version of it is that they tested gerbils in an area which contain two landmarks, the two circles here, and then the X indicates a location where they could dig for food. And they were tested over and over, trained over and over on this again, but then on a test trial, the two landmarks, the two objects were pulled apart, and then what they observed was that the animals did not search in the middle here, but they actually searched at a certain distance away from each of those objects, suggesting that they actually encoded the distance and direction from individual objects to find the food. And this, together with theoretical work that was inspired partly by this, and that includes the work of McNaughton and et al., and Jim Knierim was also on that paper, which suggested that there must be cells in the hippocampal system somewhere that actually respond to locations defined by distances and directions or vectors from individual objects. And The idea of vector encoding was also proposed by O'Keefe and Burgess based on their work, but they suggested it was walls or boundaries that were used by animals to encode positions in the open space. So the idea was there. So based on this, then Øyvind Højdal, who's a Ph.D. student in our lab, recorded from mice when these mice were running around in very simple environments like the ones we have seen already, but there was now an object, a very prominent tower-like object in the environment. And it turned out that there were actually indeed very many cells that responded to the location of the object. They did not fire at the location of the object, but they fired at some distance away from it in a certain direction. And such a cell, an example cell, is shown here. You see it has one single peak of one single area of firing, and that area is displaced from the object in a certain direction. So the typical design is like this. It starts out with a no object trial. There's no object in the circular environment. Then an object is introduced somewhere near the middle, and then the object is displaced. And what he sees, like in the two example cells shown here, is that the cell starts to express a strong field, strong area of activity at a certain place defined to the object, in this case on the north side of the object, some 20-30 centimeters away. Then the object is moved. So in this case, the object is moved down. You see the white circle here. And still, the cell fires some 20-30 centimeters north of the object. And the same thing for this cell shown here. And that can be plotted. So you could plot the firing rate as a function of distance from the object and the orientation of the object. And you can measure that on trials with objects in different places. And then you find for these cells that they have correlations between those two trials that are way beyond what you would expect by chance. So how are these object vector fields distributed? So this shows just data from a part of the data. Actually, many more cells now since I had this figure. But what you get here is still the point. This shows one line per cell, and color indicates firing rate, and this shows orientation relative to the object. And what you can see is that essentially all orientations are expressed. If it was perfectly distributed, you would have a line along the diagonal here. So this is the distribution of orientations, a slight bias towards 90-degree cycles, but actually that bias has been almost gone now in the larger data set. This shows the distribution of the distances. So you can see typically the firing field is about 5, 10, 15 centimeters away from the object, but it can be anything up to 45, probably more than, but we couldn't test beyond that because the environment wasn't really larger than that if you wanted to have the object in different places. It's not dependent on the exact type of object. So this shows 13 different types of objects, so many of them quite similar. They are tower-like. They could be prisms or they could be cylinders, but looked quite differently. And if you then use several of them or replace them, it doesn't really matter. So this is shown here for 5 example cells. So you can, for example, see cell number 2 here responds in the same way to 2 objects placed here. So there are 2 circles, and you can see it fires on the left side at a certain distance away from each of the 2 objects. So you can also see it here. This cell, 3 different objects among these, and again on the southeast side of each of them. And this one here, you can even construct a grid cell if you like, because if you put the objects in a certain pattern, you can get fields on the, in this case, on the northwest side of each of them. So this suggests that it's not really the identity of the object that is encoded, but more like positions and actually vectors, directions and distances away from any prominent object in the environment. And that even includes some that are very different, like the flat cylinder here, and even a wall like this. So these cells, is this something that has to be learned? Well, it turned out not to be, because these cells that were recorded multiple times in familiar environments, they were also tested in a novel environment, novel room with a novel object, and you can see that you get the same, exactly the same type of firing, both in the familiar and in the novel room, and there are, if anything, just very, very minor differences in the information content or in how spatially selective they are. So we also wonder is the intrinsic relationship between different cells of this kind maintained? So what you see here is two simultaneously recorded cells. So one has a field on the southeast side and one has a field on the northeast side in room A. And if the rat is then recorded in room B, well, then it all rotates for this cell. So this one goes or flips almost 180 degrees. And now you see that the field is on the northwest side. And this one also then flips by 180 degrees almost, and the same happens to a head direction cell that's recorded to the same time. So this suggests that actually the intrinsic relationships between these cells and even between these cells and other directionally oriented cells is maintained between environments. So again, this is part of the low dimensionality of the entorhinal map, where both grid cells and head direction cells, which I didn't mention, actually turn out to be more or less one map that is maintained across environments. Are these cells different from other cells like grid cells? Well, largely yes. So we calculated scores for both border cells and grid cells and speed cells and head direction cells using different criteria that we have used previously to identify such cells. And essentially what you can see here is that for example, grid scores are around 0. That means that it's not different from what you would expect by chance. And also for the head direction tuning, what you see in the middle column is the object vector cells when there's no object, and then to the right you see the object vector cells when there is an object present. And what you see in the left column here, or the left one, is the rest of the cells. So it's not really different from the population. So low head direction tuning, low grid tuning, and not really, it's definitely not more border-like activity than border cells. However, yeah, I'll skip this, but however, there is some overlap with border cells. And that's maybe not so surprising, because a border is also an object. So, I mean, how you really distinguish those, because a border is just an object that is elongated in one direction. So when does a border become an object, and when does an object become a border? I think this is not totally obvious where the boundary is. So, but using the criteria we have used to identify both object vector cells and border cells, then we do find that there's a small subset, 11 out of approximately 150 cells, that actually satisfy both criteria. And you see some examples over here. So if you look at the bottom first, you see a typical border cell recorded with no object present. Then the object is introduced here. In the middle you see a white dot here. And then the cell adopts a field on one side, just like it has for the border. And here at the bottom you see another cell of the same type, which fires along the border of this cylinder. And then when you introduce the object in the middle here, you get another field as well. But many, many don't, and most actually don't. So you see an example at the top here, where there's no object, it's a border field along one border here. And then you also see that in the neighboring cell or in the one below here, and you introduce the object and nothing happens. So what is the difference between these cells? It's not quite clear, But there are many things that suggest they are not just object vector cells are not just border cells that they are different in many ways. So what we show here is that this shows just the relationship between the orientation of firing of these cells. So this is the direction of firing relative to the border. So this is for the border cells, and you can see that they essentially line up along the orientations of the walls as expected. But when it comes to the object vector cells, which you see here to the right, they have all kinds of orientations. And the same with the distance from the object versus the wall. So this shows for the border cells, so this shows the orientation of the object vector field versus the orientation of the border field for those cells that had fields in both, and you can see there is really no correlation. You would expect bands along in parallel with the diagonal if there were. And the field distance, so this is the distance from the wall or the border. This is the distance from the object for those cells that fired in relation to both, and you can see that the distance from to the object is much larger. So this could be because of the way the cells are defined, but nonetheless they are different in many ways. So then to try to tidy up in that, then we have an ongoing work which is Sebastian Andersson's work, where he tried to manipulate the shape of the objects to make them more or less border-like. So first of all, he tried to ask whether it's the height of the objects that matters. So he had small objects and then big objects, and then put them in the same place in the same environment, and then could see that if they're very small, sometimes they don't elicit object vector fields, but consistently as they get larger. And the same when he changes the width of the objects. So this shows anything from about 2 centimeter width to 30 centimeter width, and you can see that the cells fire in the same orientation in the same way regardless. So, and he even tried to morph the objects from what we called an object originally to a border or a wall, like you see the object here getting bigger and bigger and bigger and then going back. So essentially the cell fires all the time, but this cell, which clearly is an object vector cell by definition as we had it, it still doesn't, even if it fires along most of the wall here, it never fires along the peripheral walls. So I think these cells, they have many properties that distinguish them, although it's still yet to be finally determined what is the difference between an object and a border. So finally, before I leave this topic, I just want to say that these cells have some similarities with cells that have been recorded before. So first of all, I want to mention the object cells, not object vector cells, but the object cells of the lateral entorhinal cortex, which Jim Knierim and his students have observed for many years. But these cells essentially fire at or around the object, so they're different in that sense. But the object vector cells are more similar, possibly identical, or at least more similar to what they call the landmark vector cell in the hippocampus, which are cells that also fire displaced from the object. So this shows 4 objects, and this shows the firing fields, which are, in this case, on the southeast side of 2 of the objects. But they are also different in some ways, like, for example, many of them fire only in relation to some objects and not others. And as I understood, it also took quite a while for many of them to develop, whereas the ones in the entorhinal cortex are expressed from the outside. So, and finally, just to sum up again and come back to where I started with these cells. So these cells, they suggest then that the medial entorhinal cortex may encode position in several ways, not only by a metric defined by a regular grid, but also by actually using completely different principle, a vector-based principle based on locations relative to objects in the environment, individual objects in the environment. So, and then I promised to come back at the end to another dimension, time, which is, we heard this morning that, and especially from Lynn Nadel's talk, that hippocampus is very important for episodic memory, where space has an absolutely essential role, but space isn't all. There's also in episodic memory a time component, and our understanding of how time is really encoded has not been at the level of our understanding of space. So this is the work of Albert Sau, who was a Ph.D. student in our lab, and it's also a collaboration with the CNIRM lab, which have contributed some of the data. This will be presented in more detail tomorrow in a symposium, so I don't want to steal the whole show from Albert. So I'll just present it very briefly and put it into context, and then hopefully many of you will find an opportunity to listen to Albert himself tomorrow. But let's put it in some background. So what do we know about encoding of time in hippocampus? At least two aspects that are worth emphasizing. First, we have the so-called time cells, which are the cells that were described initially by Pastalkova et al. From the Bouchaki lab, and then followed up more extensively by a series of studies from the Eichenbaum lab, which showed that when, this is then the original task, when rats run in a certain pattern, like in a figure 8 pattern like you see here, and then stop in a running wheel to run for a delay until they continue to run in the maze again, then during that delay, the cells actually fire at certain times in the interval. So this is plotted here by neuron number 1 to neuron 30 here, and then you have time in the wheel when they run, and what you can see is that these cells fire at specific times during the interval, and that it's a very orderly firing, just like cells fire orderly in space. When rats, for example, run on a linear track, they fire in a certain order when they run on the wheel, even if they don't move at all. So it is not, and this even happens when they control for movement. So this was proposed then to show that hippocampal cells can actually also express time, the very same cells that express space in other contexts. So this so-called time cells, there's a lot of attention to that now. But nonetheless, these cells, this is described across time scales of not much more than 10 seconds or a little bit more. And probably this also has to be learned. But then there's a totally different expression of time in the hippocampus. So going first back to studies again by Eichenbaum, where they showed this is a study from 2007, where rats were trained in an odor sequence memory task. But the essence of it is shown in this figure. So this is the trial lag, or the distance between trials. And on the y-axis you have the differences in the population activity. And what you can see is that regardless of where they actually find their food, the distance increases with time. So there's there's a change in which cells are active at any given time in the hippocampus that could be an expression of time. And then work from Jill and Stefan Leukeb's lab, and also a study from Siv et al. When he was at Mark Schnitzer, showed that in CA1 this is strong, yes, but it's even stronger in the CA2 area of the hippocampus. So this is where we were when Albert Sau started, but we wanted to find out more about this and also where it came from, and try to understand how such a code is expressed outside the hippocampus. And we we then directed our attention to the lateral entorhinal cortex, which I haven't talked much about at all today, but where cells are not really very strongly spatially selective, as was shown by Jim Knierim's group about the same time when we found the grid cells. But we wonder whether much of the activity of lateral entorhinal cortex could actually be explained by a role in coding of time. So what Albert did was that he tested rats in a sequence of trials extending over a period of more than 1 hour. So going from alternating between two types of environments, so it's a black environment and a white environment, so it means that the walls are either black and white, otherwise totally similar, and then alternating in a semi-random sequence from over a series of 12 trials, and then in between the resting trials or pause trials, so that total is 24 different recording epochs, and as I said, total a little bit more than 1 hour. And then he asked, how is the activity of cells in the lateral entorhinal cortex during recording over this time sequence? And first of all, he did find some cells in the lateral entorhinal cortex that are strongly modulated by time. That means that their firing rates change in various ways across the experiments. And this is not due to instability of the changes of the cells, because he showed in many ways that it is totally stable. But the cells, you see the activity of the cells is shown here for 4 different types of cells. This is across the alternating trials or sessions of black and white trials. And what you can see here is maybe a little bit difficult to see, but these cells ramp either up or down within trials, as in this one. So firing rate begins low and gets higher and higher and higher. Then next trial begins low and gets higher and higher and higher, and so on. Or it might have activity that could ramp up or ramp down over the whole 1 hour or 1 hour plus session. Or you may have combinations where the activity ramps up or down just in the black boxes or in the white boxes, and so on. So based on this, he then performed general linear model analysis, GLM, and identified the fraction of cells that had significant modulation by the various factors that he put into the analysis, which included the color of the wall, black or white, the position of the rat, or the combination of the two, or time, or mixtures of them all. And what he found, first of all, if you begin at the bottom with the medial entorhinal cortex and with the CA3, not surprisingly, there's a very strong influence of positions. You can see both here in the medial entorhinal and also to some extent in CA3, of course, and not much in lateral entorhinal cortex. When it comes to the color of the wall, quite low in all of them, but some in the CA3 and in the lateral entorhinal cortex. But when it comes to time, you see that both CA3 and medial entorhinal are quite low, but when it comes to lateral entorhinal cortex, it's a very high proportion that have significant modulation. It's about 25 to 30% that pass that significant threshold. That doesn't mean that the others don't, but they may have weaker influences. But then the CA1 and CA2, they are somewhat in between, but none of them really reach the level of the lateral entorhinal cortex. But then he asked well, these are individual cells, but could it be that those cells that don't pass that threshold perhaps also contribute to the coding? So he took a totally different approach then, looking at the whole population instead, and used a machine learning approach or a linear support vector machine to determine the contribution of time for the 3 areas, lateral entorhinal cortex, CA3, and medial entorhinal cortex. So what he did was that he trained a network based on he chopped it into 10 blocks and then trained it on 9, and then used that to predict that for the 10th one as a test case what time this actually was recorded in across those blocks of 24 epochs. And the success of that shown here. So this is in this confusion matrices here. So what you have here on the x and y axis is the predicted epoch on the horizontal axis and the actual epoch on the y axis. And then color indicates the proportion of hits. And you can see the strong, almost every case is a hit here, and very, very few were actually mishits. So you could almost all the time predict when the recording actually took place. This is not the case in CA3, as you can see here, and just weakly the case in the medial entorhinal cortex. Corrected for size of the sample. I don't want to go into that. And this is also, it was not only able to predict epochs or blocks of trials, it could even go within trials and predict the right 20-second block, or even 10-second block, or even 1 second. I mean, of course, with much lower success if it was 1 second, but it's still, you see the stipple line here at the bottom, that's the chance level. So it actually means that this representation of time was present at multiple time scales. Finally then, one could ask, is this an internal clock that is present in the lateral entorhinal cortex? So is this a clock-like thing that goes on regardless of what happens? Or is it actually dependent on the experience of the animal? And it may turn out to be the latter, which I will show in this final data slide, which again, now is a new type of task. The rat is not walking in the open field box like it did before, but now it's running in a maze in a figure 8 pattern, so alternating left and right on every second trial. What he found in this task, and also in one other task where the animals just run in a circle over and over and over and over again, is that in those tasks, if you then decode the identity of the trials, which trial the recording was from, actually the success is lower than it was in the open field. So it's much reduced in those two tasks, in the figure 8 task and in the circular task, compared to the task when the rat was walking in the open field. And you can see here there's very little activity, really very little, few hits along the diagonal here. But what was at the same time as this was lower, it's just a repetition, lower in the success of hitting the right trial, whether it was trial number 5 or 7 or 9, when it comes to when in the trial the recording was from, then it's reversed. So what he did was that he chopped up segments each time the rat passed a certain point on this track, and then he did that for every lap that the rat went through and through and through and through. And then he asked, is this from an early one or a late one in that trial? Then the success is actually reversed. So now, when in this task the hit success was much higher than it used to be when the rat was running freely in the open field. So this then suggests that the encoding of time in the lateral entorhinal cortex is not a fixed thing. It depends on the experience that the animal has. And this network, the representation of time, can actually be adapted to what the animal experiences, going from a kind of absolute, in quotation marks, representation of time where it's just running freely, to one where you actually encode time relative to some temporal landmark, like, for example, the start of the trial or passage of a certain point on the track. And that then brings me back to the person who introduced me, who was, so Mike was kind enough to allow me to show one slide from his own data. I asked him for him because Maria Monchal from his lab has done work in humans, human fMRI studies, where they actually find that have data that are entirely consistent with the data from the lateral entorhinal cortex in the rats. So what they find, very, 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 very briefly summarized, is that they let subjects view a movie, a famous TV thing that I have never heard about. But the point is that after the movie, they were asked to put on a timeline when a still image from the movie was shown. Was this early or late or whenever? And then they could measure how well they hit. And the hit rate was actually very highly correlated with activity in the lateral entorhinal cortex, no correlation with medial, and also high with the perirhinal cortex and not with the para, which and perirhinal is very strongly linked to the lateral entorhinal cortex. So this, I think this is, those two sets of data fit very well together. And with that, I think I come to the conclusion. So these are people from the institute and from the lab. As Mike mentioned, Edvard Moser participated in all of it. There are also other people who I've listed here. There's so many I can't really mention them, but for the new work, I would again then mention Albert Zhou's role, and who is going to present it tomorrow, and also for the object vector cells, especially Øyvind Højdal, and also the more recent work with Sebastian Andersson. So with that, and a lot of people pay for this, and Eurlings sponsored the lecture. So with that, then I'm done, and thanks you for your patience sitting here so long, and I'll hope you'll have a nice evening.